Hi, I'm Rafael Fonseca. You're here at another episode of Innovation in Medicine. It's a pleasure to be here today with Brian McMahon. Brian from Sparkcures. We met each other through acquaintances through the myeloma crowd, and Brian has been a caregiver, and he'll tell us about his story. He was caregiver for his mom, who had multiple myeloma, and that sent him in a quest and a trip that he obviously has been pursuing for the last several years, and just doing a tremendous job trying to make a reality for patients to have access to clinical trials. So I'm just delighted to be here with you, Brian. You and I talked recently. We worked together at Salt Lake in a meeting for the myeloma crowd. And I think as you and I agree, we see a lot of things that we need to do as a community and that we could do together as well too, to try to advance some of the ideas that you promote through Sparkcures. So first of all, thank you for being here.
Yeah, of course, Rafael. Thanks for having me. It's a pleasure.
So Brian, just to kind of set the stage a little bit as we go through our conversation, would you mind, maybe we can start just briefly with Sparkcures, and then I'm going to ask you about you and your story and, how do you get here? But why don't we do just a little bit of a nutshell of what Sparkcures is, so that then we can walk our way into, how that came to be.
Yeah, of course. So at Sparkcures, we're laser focused on helping myeloma patients in the U.S. find and connect with eligible clinical trials. And it was interesting for us because when we first started, everything was about clinical trials for us. And we'll kind of get into the genesis of that and how our system works. But what we've learned over the years is how important a myeloma specialist such as yourself, and there's a lot of really great myeloma docs out there, is and what impact that has on patients. So one of the things we've sort of changed over the past year or so is not just focusing on clinical trials, but focusing on myeloma centers and myeloma specialists. So one of the first questions we ask any patient is, are you being seen by a myeloma specialist? Because if they're not, what we want to do is try to figure out what's around them and what available options they maybe have so they can augment their care, right? And this might be putting the cart before the horse, but one of the big fears that we see is people will say, well, I love my local doc. And we say, great, you can still love him, you can still be seen by him, but let's augment the team and let's add someone in who does nothing but multiple myeloma to really, put a cap on top of this and maybe help be a coach where maybe you'll see them once a year, once every 2 years, depending on if your MGUS are smoldering, or every 6 months. That way you can still receive care locally, but you have a myeloma specialist on your team.
And let's dig a little bit into that before we go into your story, because I think that's such an important point. I think is just a point of, the reality of what's going on with multiple myeloma. As I think many of our viewers will know, things change so rapidly that it is not an exaggeration to say that every 3 to 6 months we might be doing things in a different way from what we were doing before. So there is a tension of, how do we, first of all, recognize that this changes so fast, are able to work in partnership with some of our colleagues in the community, but at the core of it, recognize that it's so hard to keep up with all of this. If you're a doctor and you're seeing breast cancer and lung cancer and lymphoma and myeloma, it really would be a superhuman feat to be able to stay on top of everything, correct?
Yeah, without a doubt. I mean, if you think about it, I'm reading a book. You gave me some great book recommendations, which, by the way, I've picked up 2 of them already. I'm reading a book right now called The Checklist Manifesto.
Mm-hmm.
And it's a community physician who talked about the amount of patients that they see and the number of indications that they have to be aware of. And it's so difficult to stay on top of this. And I think that's why for our specific purpose, we want to stay strictly on myeloma, strictly on clinical trials, right? Because staying on top of that is such a critical and time-consuming and difficult process. And oftentimes we'll hear patients come in and they'll say, well, won't my doctor tell me about this? And we hope that they will, right? And our goal is to augment the conversation with a patient and their doctor, not to suppress it, not to circumvent it, none of those things, right? So the thought is, if you're getting this information from your doc, and I think at the event that we were at, a patient had even asked, are you up to date? And we know that you and your team and a lot of these myeloma specialists are staying up to date on all the latest research, what the different bites are doing in the antibody drug conjugates and the CAR Ts. But when 80% of the patient population or thereabouts is receiving care in a community setting, it's so incredibly important to make sure that you're getting some accurate, up-to-date information. For somebody, it's not a knock against general oncologists or hematology oncologists. There's a role, and there's a very important role, but when you're coming down to it, we want to make sure that we don't leave any options off the table, right? And we want to make sure that we're really having a conversation because, as you know, we always say that every treatment decision is going to impact your future, right? Whether that's standard of care, whether that's clinical trials, everything is going to impact. So every time a patient has a line of therapy or a treatment, it could take away some clinical trials forever, and it could open some up for the first time. So navigating that and being aware of that is such a critical piece of this puzzle.
And now, one of the challenges too, as we're going to go into the clinical trial conversation, is that, as you allude, the majority of patients are cared for in the community, and some of them will have access, of course, through clinical trials, through some community groups. You know, there's notable examples of this in the United States, like US Oncology, which has a very robust network for clinical trials. But be that community or academic, only a very small minority of patients participate in clinical trials. Now, clinical trials are always an option, and clinical trials have to be offered and, should participate only if it's the right option for a person. But I think most people feel there's a huge gap between what would be a potentially good level of enrollment for care for oncology and clinical trials versus what actually happens, which, as we know, even in academic centers, is a very small minority.
Yeah, I agree. I wonder if it makes sense maybe to talk through sort of the building blocks of the clinical trial, right? So I think one of the things that we look at is the inclusion-exclusion criteria. And if that's sort of a good path, because I think that all leads into this together, right? Looking at how do you sort of plan out and understand what's currently available, what might be available in the future, and what's no longer available, and how do you sort of navigate that, right? Because I think the awareness and understanding plays such a critical piece into what you're saying there, right? Because oftentimes people may look at it and they'll say, well, I still have options left. And what we see is that's awesome. That's a great thing to have standard of care options, right? And there's sometimes that it makes sense to pursue that. And then there's other times where we might want to be considering a clinical trial at this piece. And I guess the question before we get into that for you, when you're in a clinic having a conversation with the patient, how do you help weigh the pros and cons, or how do you help them look at and sort of, how can you put up a clinical trial versus standard of care and help them work through this to decide which one might be an appropriate option?
That is a great question. And I'm glad you asked, because I was thinking as you were talking that maybe for the, setting the stage for what we're going to be discussing with clinical trials, there may be some members in the audience that don't necessarily understand the various steps. But let me first address your question and then talk a little bit about the blocks. So first and foremost, I hold this very deep belief that when you're in an office with a patient, you're seeing a person, the only responsibility is to do the best there is for that particular person. And obviously, no one holds truth 100%, but in your best professional opinion, what would be the best course of action for that person? Now, sometimes the best course of action might be to say, listen, we do have this standard of care treatment, and what we would like to do is to propose you receive such a treatment, a combination, a drug, whatever. Now, into that conversation, so as we ponder on what's the best option for a patient, sometimes that may be including the participation in a clinical trial. And people will sometimes think, well, clinical trials aren't there just like a last option thing? And the answer is absolutely not. So it may be that I'm seeing someone for the very first time, someone who is newly diagnosed with multiple myeloma, and there may be a clinical trial that fits exactly to what they need. And usually, a trial that would be used for someone who has newly diagnosed myeloma usually would be permutations of things that we already do, or perhaps the addition of a new drug that seems somewhat promising into existing regimens. And speaking of those blocks, we do have essentially 3 levels of clinical trials. We, of course, have the phase 1s, where you gradually traditionally increase the doses of medications when people are looking at toxicity of particular drugs, and I'll get back to them regarding myeloma in a second. Once you go through that process of understanding the safety and perhaps some toxicity aspects for the drugs, people go to a phase 2, where you test the drug and you try to estimate what fraction of patients respond or show benefit from that particular drug. And phase 3s are when things are compared to the standard of care. And starting from your question, when we do something, when we think about offering one of these clinical trials, we have to hold the belief that the clinical trial provides an option to a person that would be just as good and hypothetically potentially even better than what they currently are receiving for their treatment. So, why would I say to a person, listen, you have drug A plus B is your standard, but we have this clinical trial that compares A plus B plus C. The reason we're doing it in a trial is we just don't know if adding C makes sense or not. But the reason it's there is because there's some indication that C might be beneficial. There's some indication that C has worked on its own, has added to combination A plus B. Likelihood has gone through phase 1 and some often like phase 2 trial in such a combination. So that's how we think about clinical trials. And sometimes they will bring options to patients that don't exist in any other way. I mean, a great example for this would be, CAR T cells. I just yesterday saw a patient for whom there's a CAR T cell trial option that is not available in any way that I could prescribe it, of course. It's not available through standard of care. So that's kind of how we go through the thought process of those trials. But it's I try to simplify something that is incredibly complicated, of course.
Well, and so when you talk about the building blocks, right, and we look at phase 1, phase 2, phase 3, one of the challenges on phase 1 is we'll have people coming in saying, I'm not willing to do a phase 1 trial, right? And at the end of the day, if someone's not comfortable, don't do it, right? This is your personal preference. One of the things that we like to impress upon people is have a conversation with your doctor. Make sure that you're coming towards this with all the information, right? So first in human, very different than maybe a drug that was used in melanoma or another indication like one of the leukemias or lymphomas, where maybe more patients have had that, a little bit more safety is understood about that, right? And maybe there's dose escalation, right? Where they're elevating the dose on these different cohorts versus dose expansion, where we've determined the dose that we want to move forward, and now more patients are going in there, because it's a nuance that's difficult, I think, for a lot of people to grasp, but it's an important factor for you as the physician looking at weighing those options for them, correct?
Absolutely, and I'm glad you brought again up the issue with the phase 1. So phase 1s in general are not used for the most part for newly diagnosed situations, but they're usually a venue to bring new compounds or new ideas. But I mean, the world has moved forward quite a bit. If you go back 40 years, phase 1, we're just, throwing random things and hoping they would work because there was some study on some animal model or perhaps some in vitro testing that would perhaps propose that a drug, at the time, mostly chemotherapy, might be effective. Nowadays, the numbers are actually quite better. There was a NASH abstract, I believe, a couple of years ago that looked at overall response rate in phase 1 trials for myeloma. And if I'm not wrong, I think they reported 40%. Which means there's a tremendous amount of thought process that now goes into, what we're testing in phase 1. So, for instance, if a company were to develop a new antibody against CD38, such as daratumumab or isatuximab, even though they're going to have to go through phase 1, we know there's a high likelihood that an antibody like that would work, right? So it's just refining what we might understand about the toxicity. Or another example might be we're doing a phase 1 clinical trial right now with a bispecific antibody, which again works like in many ways like a CAR T cell. But the reality is we go into this trials thinking that as soon as we start hitting the right dose, we're going to hopefully start seeing some activity. And that's what has been seen, for instance, with bispecifics, reports of which already have been presented.
Yeah, and it's a really important point that you bring up there. I mean, if we look at the data coming out of the CAR Ts, right? So obviously the front runner of this is the BB2121, right? A BCMA targeted CAR T. And if you look at the data coming out of their KarMMa-1 or the initial phase 1 study, right? It's really incredibly impressive for a phase 1. And I think the challenge is the level of expectations, right? When the press picks up on something and you start talking about a potential cure, and then you end up getting between a progression-free survival median of 12 or 15, all of a sudden it feels a little deflating. But I thought that was incredibly, incredibly encouraging for such a, for a patient population with between 8 to 23 or 25 prior lines of therapy, right, to be seeing some of the results. And probably the same thing, you can talk more about this, is the AMG 420,
Sure.
specific, the bispecific antibodies that are BiTEs. And, actually, if we can pause there for a second, I'm curious. So are we splitting hairs by saying bispecific antibodies and then bispecific T cell engagers? How do you guys look at that, and how do you communicate that to patients? I'm curious.
It's a great question. I usually use the term bispecific, and I know that involves a lot of things. There's different molecules like the BiTEs and all sorts of other strategies. The way I describe it is these are the double-sided Velcros. So this is a Velcro that on the one side brings the myeloma side and the other one brings the T cell And I think you've heard me say, I use the analogy of the, the scorpion and the frog for T-cell engagers. You have that story of the frog that needs to cross the river, and the scorpion goes on top, and that's a T-cell, that's a scorpion, right? So I think semantics will evolve as technology evolves. So I use indiscriminately right now the term, bispecific. I think we'll refine that as, first of all, as products advance, as we understand better their mechanisms.
Well, 2 points there. At the myeloma coaching event where we were last week in Salt Lake, you had a presentation and you referred to the bispecifics as the matchmakers, which I thought was such a really great analogy, right? We're looking at, for some reason, someone's T cell is not picking up or able to identify or target that myeloma cell as a bad actor, as it were, right? So now we have, this antibody or this molecule that's able to grab hold of the myeloma and the T cell to bring them together to activate, to act as a matchmaker, which I really appreciate. I thought that was a great.
That's the other one I use, matchmaker, and the double-sided, Velcro. That I think brings a point home, which reminds me of something. So let me ask you something else. So one of the things that struck me when we had the conversation in Utah is that because of what you do in Sparkcures, and I want to ask you a little bit more about this, there were some points where I thought, hey, wait a minute, Brian knows more about the clinical trials than I do, because you're curating this data, and you tell me, I'm sure you're aware of all of this, but as it always happens, it's so hard to keep on top of things. So I have an automated email that comes to my inbox with every single publication that comes out from myeloma. Reading that email is a task because there's so many publications that come forward. So why don't you tell me a little bit more, since we put the blocks there for the clinical trials. So what is the vision for Sparkcures, and how is that going to help patients?
Yeah, would it make sense to maybe kind of talk through the genesis of that, and that sort of leads into where we're going?
Please.
Really, this all started. So back in 2005, my mother was diagnosed with multiple myeloma. Now, the interesting thing about this was, at the time, she had 2 high-risk mutations. So at the time of her diagnosis, they gave her 30 days, even with treatment. Now, my mom was a very atypical myeloma patient because she was an oncology nurse with 30 years experience. So she had been treating myeloma patients for years. She was on the local board of the Leukemia and Lymphoma Society. And at that point in her career, she was actually on the Neupogen team at Amgen. So, so she was actively involved with blood cancers across the board. So when she was diagnosed, not only did she have the wherewithal and the industry sort of knowledge, right, as an oncology nurse to sort of navigate this, she also had what we call the Rolodex from her time as in the pharmaceutical side of things. So, she was fortunate enough to be able to pick up the phone, call the head of the UPMC Cancer Center, Dr. Stan Marks here, and say, Stan, what are we going to do? And at that time, it was, University of Arkansas with Dr. Barloghi and Dr. Trico. So within 2 weeks of her being diagnosed, we were on a plane to the University of Arkansas. She ended up enrolling on the Total Therapy 3 clinical trial protocol, which is the tandem back-to-back transplants. Now, unfortunately, after less than 2 years, my mother actually passed away from her disease. But what we like to say is that we were expecting 30 days and we got 2 years out of this.
Right.
And, it really opened up my eyes to the power of clinical research. And what I like to say is that my mother lived for 2 additional years than we would have thought, because of all the myeloma patients that came before her, because of all the research that had gone into better understanding myeloma and providing better therapies, right? I mean, melphalan and prednisone, and all of a sudden she was getting back-to-back tandem transplants. She was actually diagnosed before Revlimid was even approved. Revlimid was approved in December of 2005. So she was diagnosed before Revlimid was even a standard of care therapy.
Wow.
And the challenge that we saw, so in the span of about 10 years, I was the primary caregiver 4 different times. My mom was the only one to get into a clinical trial. My business partner passed away from neuroendocrine cell cancer, which is the same cancer that Steve Jobs had, so a variant of pancreatic cancer. My father-in-law passed away of pancreatic cancer, and my grandmother had cervical cancer. And we were unable to get any of them into a clinical trial. My mom was the only one. And for me, what happened was, this is around 2012, my business partner passed away. And 2 months later, so we ended up making the tough decision to shut the company down. And I've always been a startup guy, so I hadn't really been on a true vacation in 6 years. And my wife and I went off and we were on a vacation. We got a call. Her father was in the hospital up in Boston. And it turns out he had pancreatic cancer. Within 2 months, he passed away as well. And, Raphael, I thought, I was looking at this and I thought, well, I'd always been involved with cancer. You know, my mom was a big supporter, like I said, of the Leukemia Lymphoma Society and the, you know, Oncology Nursing Society, ONS, and all these different functions. And I always thought I was going to do something to help out on cancer. I was always drawn to that. We did a lot of fundraisers, a lot of different events. And I thought my father-in-law was 48, my mom was 55, and my business partner was 62. And I thought, you know, 40, 50, 60 isn't guaranteed, right? We don't know. I hope that I'm going to see those, but I may not. And I thought, if this is something that is so critically important and I'm seeing the struggles firsthand, why am I waiting until 50, 60, 70 before I try to give back or do something? And that's when we really decided to pursue this. And the thought process was that you shouldn't have to be an oncology nurse or work for a pharmaceutical company or be a myeloma doc to be able to navigate and pursue potential clinical trial options as a patient, right? When Jenny Ahlstrom, right, from Myeloma Crowd and Healthtree, tells the story when she was looking for clinical trials, she went on clinicaltrials.gov. And I always say this, it's not throwing stones. This is a really difficult problem. But what we found is that a lot of the information on clinicaltrials.gov was either outdated or inaccurate. And she ended up having close to, I think, 350 results. And the question becomes, what do you do with that, right? If I'm going to go see you, right? If Dr. Fonseca is my myeloma doc, am I going to print out 350 trials and bring it into an office appointment and say, all right, doc, what are we going to do here, right? That just doesn't really work. So the question became, how can we use technology and how can we better understand what is a trial looking for and what is a patient potentially looking for? And, you know, just like the bispecifics act as a matchmaker, can we act as a matchmaker and make sure that the correct options are placed on the table so that you can then have that conversation with your doctor? And that was kind of the genesis of Sparkcures.
You know, and as you were chatting, I was thinking, so Brian, you know, you self-describe as a startup guy. You're you know, technology savvy. This was 2005, right? So there could be someone in the audience that says, well, listen, it's 2019. I'm probably just going to Google. I'm going to find what trials are available, right? And I mean, the reality is that's going to be very, very hard. You alluded to, of course, you know, the central repository that exists for clinical trials through the government. And again, not throwing stones. But I always think of, you know, this paradigm of, you know, the data, information, knowledge, and wisdom, right? So the data and the information is out there. And I think how you contextualize that and whether that's the right thing for me or not. And one of the things you try to solve with Sparkcures is, which brings the layers of the real world, the practicalities. Okay, is this something that I have access to? Is this something that would be practical for me to consider because I have some support system, etc.? So I think it's really quite a task. I mean, we do have the tools, and you're converting these tools into practical solutions for patients. But I can only imagine in 2005, which sounds like we're talking about the, you know, prehistoric age. But the reality is, in the digital world, we didn't have any of this, at least not implemented in a way as you do now.
Yeah, you know, so there's a couple things there, and I like the way you talk about that. The way we think of this as a journey, right? This is not a moment in time. So I think there's a couple differences for how we approach this. Number one, not a moment in time, right? People come and say, well, I look on clinicaltrials.gov, my doc keeps me up to date, and we go, great. Then keep doing if what you're doing is working, you don't have to go through us, and that's okay. We're not going to be for everybody. We understand that. And the thing is, leave the door open. If you ever need us or want to throw some ideas around, we're here, right? But the way we look at this is it's a constantly changing you know, situation. It's a constantly changing landscape. And part of the challenge is, think about patients 5 years ago who had a prior allo, right? Who went and had an allogeneic or donor stem cell transplant. Now, I know it's fairly uncommon, but for any of those patients, they didn't know that it was going to make them pretty much ineligible for most of the CAR Ts or most of the bispecifics or the antibody drug conjugates. So treatments 5 years ago are coming back. And the question becomes, how do you navigate that? How do you navigate if you're non-secretory, right? And how do you navigate, how do you search for something if you don't even know that it exists, right? And that's part of the challenge with the repositories that are out there, is that people aren't even aware that these things exist. And even if they can, how do they get matched to themselves, right? And, you know, we can deepen, we always talk about awareness, understanding, and then action. Right? We want to help first let people know that clinical trials are an option. We're not saying that it's the option that you should do every single time, but we want to make sure that people are aware that it's out there and the important role that the clinical research and development plays. Then once we can make them aware, we can help them understand, what does this actually look like for you, right? So we can break these things down and say, well, maybe you're newly diagnosed, or maybe you're not ready for a treatment clinical trial right now, which is totally fine. But there's a lot of really interesting observational studies. There's still ways that you can participate and help advance research and understanding. And some of these you can even do from what we say you can participate from home, right from the comfort of your couch. So, understanding these things helps you to take action to say, well, maybe I don't want a treatment trial just yet, or maybe what I'm on is working. The other side of this, one of the best descriptions, and I think I shared this last week, was we had a patient that described working with us or connecting with us by saying, I feel like I'm a frog on a lily pad. Right? He said, my thing is I want to make sure there's always another lily pad ahead of me that I can jump to, right? Because with myeloma, when that relapse occurs, the worst time to go looking for a clinical trial is when you need one, right? That's a really difficult conversation to have with people when their free light chains are doubling or going through the roof, and they're saying, my doc said I have to find a clinical trial. What are we going to do, right? And we'll help, we'll do what we can. But our thought process is when you're feeling good, when you don't need a clinical trial, let's be discussing, let's talk about what's around there. And I think one of the things that you brought up was not only do we help you understand what your options are, but we do it on a personalized basis, right? So travel is a big thing. Now at Mayo, I'm sure you have people willing to travel quite some distance to come and see you. So what we actually did was we found that most of the things that we do are based on our time as either caregivers or the feedback that the patients and the caregivers that we work with are providing to us. And we got a lot of calls from people saying, my brother lives in Colorado, or my cousin lives out in Los Angeles, or my sister's in Seattle. And they were saying, this is a place where I can go and stay. I don't want to have to keep changing my home location to explore this. So what we actually launched was what we call our centers of support. So inside of our system, you can actually add in 4 different cities around the US where maybe friends or family, your college roommate, any place where you could stay for a lengthy period of time in order to participate in a clinical trial. And we actually showcase that. We'll break those out into different buckets to make that a little bit more easy to navigate, right? So we could say, well, here's the Mayo Clinic in Scottsdale. Here's the bucket for that. Here's the bucket for Tampa. Here's the bucket for Los Angeles. And that way you can start to really get a sense of what you might be eligible for now or maybe eligible for in the future, and start to prepare for those conversations that you're going to have with your doctor.
Yeah, and that's so important because, of course, cancer affects everyone, and there's some people that may have the means to travel and not be worried. But in many, many patients' minds, they're going to think, gee, I wish I could participate here, but where am I going to stay? How am I going to afford housing and food and time away from work and all those things? So I think what you do with those centers of support is really something of benefit to patients. If you don't mind, and this might be a good point to maybe talk a little bit about, how does Sparkcures work? If you could, you could tell the audience, and I've seen the presentations, but if you could walk us through, and then we'll put some links down as well too, and then we'll put some of the materials into this video as well too. But walk us through how does that work, and what are the key elements of the Sparkcures approach to finding a clinical trial?
Yeah, it's perfect. And I was thinking as I was explaining that, it's kind of hard without understanding the underpinnings of what we're doing. So it's a great transition point. So every clinical trial has what's called inclusion and exclusion criteria, right? So really just a fancy way of saying, here's the things that are going to be required of a patient in order to participate. And then here's the things that we won't allow, right? So if a patient has had a prior allo, or if they've had a prior treatment. So there's kind of high level, inclusion, exclusion criteria. So one of the most important that we can talk about is measurable disease, right? So you're going to have to, for the most part, most trials are going to look to see how they can establish the level of disease that a patient has. So most notably M-spike, whether that's serum, whether that's in the blood or whether that's in the urine, or free light chains. Those are the 2 most common. The challenge becomes, what happens if you're non-secretory, right? What happens if you aren't expressing or secreting those proteins, right? Or what happens if your free light chains are in normal range, right? So some of the trials will allow for bone marrow plasma cells. Some will allow for a measurable plasmacytoma, and a very few will allow for quantifiable immunoglobulins. Very few of them will allow for that. So what we do is we look at, how do you measure disease? What's the diagnosis? So are you smoldering? Are you MGUS? Are you active myeloma? We look at the status of the disease, so newly diagnosed, relapse refractory. And we also look at your labs. So these are all things that play a role in determining. Some things will say, well, if you've had prior daratumumab, you're no longer eligible. Or if you've had prior DARA, you're now eligible, or you're only eligible once you've had a CD38 antibody, such as daratumumab. So what we figured out was it becomes pretty trivial if we can understand what a trial is looking for from the inclusion-exclusion criteria, and we can get enough information from the patient, we can build a system that will look at every single inclusion-exclusion criteria and will match a patient based on those things. Now, we're not here to say this is your number one trial, this is what you should do. We're just saying based on the criteria, these are the trials that you might be potentially eligible for, right? So we call it high-level pre-qualified, right? Because there's always things such as echocardiograms or some things with your liver that we don't go into. So there's really low-level pre-screening things that have to occur. But for the most part, what we want to do is figure out at a high level, are you potentially eligible? And how can we bucket these things out so you can talk to your doctor?
One of the things I was impressed, so this is great. This is a great approximation of, as I'm thinking about it, options, right? This is a very good approximation of what your options might be, but you even had a layer there of doctor review, right? So there's some things that may be open to some interpretation. I think for those of those who are, are hearing us, the eligibility and exclusion criteria, there's usually no leeway. It's not something that, well, in this case, we won't consider that because of regulatory statutes. We have to fulfill those inclusion and exclusion criteria, but some of them have some room for interpretation or some vagueness. So, and you did include a doctor review as well too, which I think it's a very important aspect of this process.
Yeah, we don't think it's our role to make those final decisions. That's your role. That is the doctors, the treating physician is a myeloma specialist. That's what we're going to rely on you for. So the way we look at this, we actually, our system is very conservative, and we built this into the very beginning. So let's say that a trial will require an M protein, an M spike in the blood of 1 grams per deciliter, right? If a patient is at 0.8 or 0.9, we don't want to say that they're not eligible. That's not for us. We don't know how quickly they're progressing. We don't know what that's looking like. So we actually build in a margin of safety of every lab evaluation that we do. And we can actually set that on a trial by trial basis. So for instance, if we're talking to you and you have a trial and you say, well, if they're up around 0.7 or 0.8, let's pass that through and do our doctor review. We'll put that into a patient's potential bucket, but we're going to say, this is a doctor review. We're not totally sure. You're going to need to have this conversation. And the only time that we're going to say a patient isn't eligible is for things that we empirically know. So if they've had a prior transplant and they're not allowed to have had a prior transplant, if a prior treatment that they've had is going to exclude against them. So very specific things that are going to knock them out. That's the only way that we're going to say you're not eligible. Everything else we want to leave up to the docs. But very specifically, one of the conversation points that we've heard is a lot of times people look at this and they'll say, well, you're either eligible or not eligible for a trial. And we don't think that totally works, right? Because what we did was we created a not yet eligible bucket, right? And we've talked about this before, because if you're a patient and you've had 1 line of therapy, but the clinical trial requires at least 2, it's not that you're not eligible, you're not yet eligible. And it might seem like splitting hairs, but it's important for us because then we can put it into a future bucket and we can help you understand here currently is what you might potentially be eligible for, but as your treatment and as your labs change, these are the trials that may open up. And the nice thing is this is a very fluid and dynamic process. So all the buckets that we use inside of our system are updated every single day, or they're updated every time you update a lab value or any information on your profile. And it's really kind of cool. One moment for us that was really great technological moment is that it used to take us 3.5 minutes to evaluate 1 patient against every clinical trial, right? We have millions of points of data. Think about every inclusion, exclusion criteria and the hundreds of trials and everything that has to go into that. So it used to take us 3.5 minutes. And we spent a lot of time earlier this year, and we now have it down to about 1.5 seconds for every patient and every evaluation. Go through that. So if you're in there and you get new labs and you put that in there, 1.5 seconds later, all your trials are going to be updated, and things that maybe were not yet eligible, depending on that lab change, are going to go into your currently eligible bucket, right? So we're able to do this very quickly. And the nice thing, if sort of segueing into a lot of the work that we do with Healthtree, we integrate very closely with Healthtree. And one of the nice things about that is as you're updating your labs, or as your labs are being updated through the Apple Health Kit integration, right? So directly from your institution, All those things are occurring in the background, and every single day we're running and rerunning, and we're able to help people stay on top of this. So we kind of look at it as an insurance policy, right? You don't have to worry about going out and keep saying, what are the trials? What are the trials? We know what they are, and we're able to sort of differentiate them and filter them out in ways that you can better or more easily understand them.
Let me just put a quick comment there for those in the audience who may not be familiar. So Healthtrees is a tool that was developed by the myeloma crowd, where patients go in and directly enter their information, and this is baseline information, their demographics, but then it goes into some of the details of myeloma. You can go actually quite sophisticated to put in your genetic markers, prior treatments, etc. So it is a large data set, a large repository of information that will be used for identifying patients' characteristics that are unique, connecting patients that are similar, doing research. But one of the most beautiful aspects of the Healthtree, in my opinion, is that it will link with the possibility of clinical trials. So if patients are already entering all that information, plus they have the linkage, as you state, through their institutions to some of the blood work and some of their other health information, they can be linked and matched to this clinical trial. So if someone has perhaps an interest, they already have that possibility of doing that through Healthtree and going to Sparkcures. But one of the interesting things, which I think is quite different, this is very dynamic. It's not like you tested once, I was not eligible for anything, and then I'm done, right? So it changes, and not only changes, but it anticipates, which is what you're saying. The word not eligible yet is essential. Funny thing is, I mean, this is intuitive. We as humans know, Mrs. Jones, she needs to have a little bit of a higher M spike to participate. But on the day-to-day, when people are running in the clinic, they don't think about those things. Is there any trial that Mrs. Jones is eligible? No. Well, sorry, there was no trial, and life moves on. And what you bring into this is the anticipation of what might be next for that particular person, which again, it just occurs to us intuitively, but it's very hard to put in practice.
Yeah, and that's one of the things we love, and we're actually starting to work with centers from around the country. And part of that is, how can we, if somebody says my home center, where I'm receiving treatment is Mayo, how can we help them understand each month what that's looking like, how those things are changing, so that maybe when they see you 3 months from now or 6 months from now, they're already aware of those clinical trials, and we can help sort of skip ahead that conversation, right, once they get in there. So we really look at how can we keep moving that awareness along. There's some people that we help for years before they ever get into a clinical trial. That's not a problem, right? It might take 2 or 3 years before one comes up that actually makes sense for somebody. But when that time comes, we want to make sure that we can help somebody move as quickly as they can. And, and speaking about sort of the Healthtree integration, I think one of the things that I'm really happy about on that side is, if we think about this, so a lot of people use Myeloma Crowd to stay up to date on different things that are going on, right? So they're reading articles and those different things. So as part of this integration, if you have a Healthtree account, when you're reading through those Myeloma Crowd articles, let's say something comes out about bispecific antibodies. Well, we're starting to put a link at the bottom of every one of those articles saying, learn more about bispecific antibodies. And what we can do is, we call it deep linking. So you click on that link, it takes you into your Healthtree account, into the clinical trial section, and all of a sudden you just read all about bispecific antibodies, and now we're showing you all the ones that are near you that you might be eligible for, right? So now we can take people through that, again, awareness, understanding, and now they can potentially take action, right? They don't have to go out to another website and try to remember, well, how do you spell bispecifics, or is it bispecific T-cell engager, or is it any of this alphabet soup of drug names trying to remember? We're going to help, simplify that process and get them in there so they can take a look at it and then follow up if they want, or just keep it in the back of their minds for some future date, right? So, but it really helps people to stay on top of all these different options.
Yeah, and I think using your lily pad example, especially when you need the next clinical trial, time is of the essence, and this time it's quite burdensome. If you had to think, okay, I need to find a clinical trial, and by the way, I need to consider where I can stay and whether I'm eligible or not, and how am I going to pay for my travel to 3 or 4 centers, and my caregiver has to take days off of work and all those things, it becomes quite burdensome. Not doing it, just thinking about it, because then I need to sit down, I need to spend the time, but at the same time I'm going through the burden of, gee, it's challenging, my myeloma has come back, etc. So I think that's where Sparkcures provides a lot of value, in the sense that it's it's something that just shortens that process. And by the way, I should mention the way you have it in Healthtree and Healthtree as well too are options that are free for patients. It's just a service, right? They're not at this time. You will be working with centers, but at this time, patients in the Healthtree, that's all run by the myeloma crowd. Is a not-for-profit organization, and it's just something that's available for them there.
That's right. And it's interesting when we think through, if you look at the way that we're approaching this, especially on the Healthtree side, it just makes a lot of sense, right? Because you can get in there, you understand. I love their example of the twin machine, right? So how can you understand people that are like you and different treatments and different therapies that they've had and how they've done? And we know that everyone's myeloma is different, right? There's not that, unfortunately, that yardstick that we can say, but we can start to glean and understand more about this disease by doing this. And the beautiful thing is you can go in there, take a look at all your labs, keep all this information, look at standard of care, look at clinical trials. So we're really putting that back onto the patient. So I think there's this, this surgence of people really taking ownership over this, right? And being true partners with their doctors and being able to come up there. And again, if people are seeing doctors where they're talking through clinical trials and they maybe they don't think that they need this, that's okay, right? We're going after the majority of people that don't have access to this information and really trying to do a better job of simplifying it, laying it out there, and making sure that we're helping to augment those conversations that they're having.
Which I'm glad you mentioned. I should say that, if someone is listening in the audience and they go, wow, this sounds like a great tool, but listen, I don't know computers. This is as simple as it gets. Whether, you do that directly or you go through Healthtree, you just, put your basic information. It's going to walk you through the process, which is really important. Obviously, I think we've gone through a transition. There's a lot of people right now who are late stages of life in their 70s and 80s that are actually quite savvy with computers and an iPad, etc. Right? And they can enter that information. We're not talking about anything very sophisticated, and we understand there won't be, some people need more assistance, but this is actually quite, the interface is very, very simple.
Well, an interesting sort of anecdote there is that when we first started Sparkcures, we didn't even think we were going to have a web-based tool. We thought it was going to be all phone-based. And so the way I started out, I actually was calling support group leaders from around the country saying, here's my experience. My mom passed away from myeloma. Here's the challenges that we've seen, and we think we can do a better job. Do you want to come on and kind of help us out? And, everyone was so tech savvy. And I think what I related to is Facebook and sort of an older generation wanted to stay in touch with their families. And, there was one patient in particular, and her name's Sheila Miller. She's one of our patient advisors. She's an amazing person. She called up one day. She was having an issue on the site. And we had said, we'll give you a call back and we'll walk you through. And she said, honey, I have my iPad here. You just send me that link and I'm good to go. Right. And we thought, okay, like people are really, really comfortable. But, speaking to that point, if they're not, they can always call our phone number. In fact, we have people that don't have accounts or don't access it through the computer. We call them and we check in every 3 months. And sure, maybe that's actually a point to make here. So we don't look at this as just an account that you create and then just sort of let it sit out there. Once we connect with people, we're actually doing this on a monthly, on 3 months, every 6 months, every year. So there are MGUS patients that we call and check in every year. How are the kids? Go on vacation? Everything good? Awesome, right? But then when it becomes more acute, we might be connecting with somebody once every couple of days or every day as we're trying to navigate through that process of getting them to this center or that center, or, clarifying some questions before we get somebody to a trial location for screening. So we really try to bridge both of those to say, if you're tech savvy and want to do it, we look at it as kind of a choose your own adventure. You can go into our site, you can print it out, you can take it into your doc. We give you all the coordinators' numbers. If you're willing to take that on and give those calls, great. If you're not, we can be there to support you and help you through this as well. So we really try to cover all ends of that spectrum, or, based on someone's specific needs.
Absolutely. And one of the things that struck me, when we were in this meeting, they were talking about the myeloma crowd, some of the links they had with Ancestry, and they had, I can't remember, they talked about a leaf that was moving around. Yeah. You remember that? And it seems to me that, both sort of groups are, come to the realization is the real-time feedback that really is what brings also value to this, because it's not the one-time interaction. So, as you keep going, it keeps you updated. The other thing that I was thinking, this aligns very well with this whole new concept of the patient will see you now, right? So medicine has been incredibly asymmetric throughout history because there was a body of knowledge that doctors had to memorize, and then which was mostly non-accessible to non-physician members of the community. And there's no doubt there's expertise and there's knowledge and there's all of that, but patient empowerment has taken a major role in how medicine is moving forward and into the future. And this is just one more example where a patient can become incredibly self-valuable because of what they do and how they seek these opportunities. And they might bring things to the table that even the most expert of physicians might not be considering at the time.
Yeah, 100%. And this isn't meant to sort of, again, this is never meant to throw stones. We've had a lot of instances where, we've helped provide options to patients. They've taken to their doctor, and something has come back like, well, you have plasma cell leukemia, you're not eligible. Well, and we spoke about this, I think, last week as well. Well, there's a difference between active plasma cell leukemia and history of plasma cell leukemia. And there's a lot of times where, you just have to be your own best advocate, right? Because what we found is that if we can empower people, they're able to question back and make sure. And there's sometimes that it doesn't make sense, right? There are sometimes that they'll say, well, here's why you're not eligible. But we've had that overturned in some cases where, someone looking at that saw plasma cell leukemia, but didn't necessarily see that it was active plasma cell leukemia, right? So if you have less than 20% circulating plasma cells in your peripheral blood, you don't have active plasma cell leukemia. And we've actually gotten patients into trials where they were first denied at that center saying, well, you're not eligible, right? And I don't say that it either as a pat on our back or to, say anything negative against that. It just takes everybody looking at this. And at the end of the day, the more you can advocate for yourself, the more, informed you can go in there and really start asking questions. So if your doctor says, well, you're not eligible, right? The first question should be, why? And if anything changes, could that change my eligibility, right? Because then we can start to piece this together, right? And better understand. So I think that it's such a huge empowerment, right? And there's that shifting landscape of saying, it's okay. I love that the patient will see you now, but being able to say, Dr. Fonseca, well, what trials here at Mayo? And if I'm not eligible or not yet eligible, what has to change, right? Does my M spike need to go up? Do I need to see a change in my free light chains? Really helping to understand what things need to occur in order to look at a change in treatment or to be eligible for a clinical trial. And the thing we always say is, if patients do that, they can come back with that information, and that can help us to reevaluate our trial matches for them, right? So if there's a specific reason, we can go ahead and we can kind of tinker with that algorithm, or we can take a look at those trials and provide back different matches based on that feedback. So it's a two-way stream, right? We want to help empower them, just like we want to take that feedback and have that empower the matches and kind of keep that circle going full force.
Yeah, no, and it reminds me of a recent interaction I had with a patient who had a significant problem upon exposure to cold. It's one of those cryoproteins, and we were struggling as we were contemplating the idea of a stem cell transplant, how we're going to do the cryotherapy. So, many of in the audience might know, so patients put ice chips in their mouth to prevent some of the chemotherapy reaching the mouth. So we were in a conundrum, and the patient actually brought forward an option of removing the protein through what's called plasmapheresis. And to be honest, I had not really considered that, but she told me, my group in Facebook told me that someone did something like this in another institution. I thought, well, that's brilliant. And of course we did it, and the patient could get, the cryotherapy just as well. So it was something that, in this case, there was an option that was brought forward because of this patient empowerment as well, to which I thought made perfectly good sense. So
collective empowerment, right? Collective
absolutely.
Empowerment. That's why I love the Facebook groups, and I know Healthtrees is working on augmenting, some of that side of this, but, there's a lot happening in these walled off silos, right? So as we're able to come together as a group and say, well, what are you doing for neuropathy? What are you doing for X, Y, and Z, right? And being able to share that, different things like this happen, right, for the collective good, and then how do you share that and keep moving that forward is such an critical, piece of that.
Right, I'm mindful of your time, and I know that as you coming here, you announced that there were a lot of questions that we got from patients as well too. Maybe we should try to tackle some of them too, so that I think they were really curious about what would happen after this conversation.
Yeah, there's a lot of questions, and I'm going to try to, based on time, try to figure out. And I kind of wanted to move through this in sort of an MGUS smoldering into myeloma,
Please.
kind of take that through the journey. I think number one, the question is, if you're looking at a patient, if you're seeing a patient who has MGUS or smoldering, how do you go about deciding if a clinical trial is appropriate, right? Do you go off a risk stratification on looking at treating versus a watch and wait? How are you guys doing that in the clinic right now is a starting point?
It's a great question. Historically, it was a badge of honor to be conservative when it came down to not treating early myeloma, and there was a good reason for that. Historically, they were not very good treatments. Then the question, and we might say, well, listen, we don't want to treat too early. But I do remember distinctively in 2002, Dr. Fisher, who's a lymphoma specialist at the meeting we were at the NIH, said, you guys say you don't treat too early, but are you sometimes treating too late? For me, that was a moment that I thought, yeah, but he probably is right. So the community started thinking about this. A new criteria looks now towards earlier signs of clear and impending evidence of progression for myeloma. So then the question was rephrased, okay, do you want to wait until something happens? Do we actually need to see, for instance, the development of a bone lesion, creating a risk for a fracture or a fracture itself? So everyone said, no, we don't want to get there. We wouldn't do that if we're dealing with, for instance, breast cancer. So the needle has been moving backwards towards the treatment of people who have an imminent risk for progression. So this is specifically to smoldering. I think for most MGUS, there is no treatment that we provide, and it probably would be hard to find, at least mathematically, a justification that I can think of, absent some complications, such as could be, some MGUS have an abnormal protein that will deposit, and that's what's called a light chain amyloidosis, or, number of other conditions where it can affect the kidneys or the POEM syndrome. But for the most part, MGUS won't be treated unless the protein causes a problem. But in smoldering, there's a very strong interest right now in teasing that out. So the risk, of course, is coming into play, the percent of cells. I think many of the systems that stratify for risk for progression look at actual burden. And I think this is just a, probabilistic, a stochastic thing. So the more cells, the more likely you are to progress, whether that is because of the M spike, because of the percent plasma cells, because of the elevated free light chain, what have you. And I would say we don't have a perfect system right now. None of the markers can tell you 100% who will progress or not. I think the one thing that we are missing, sorely missing in myeloma, and has been explored in some recent research, is the dynamic aspect of this. And this has been used, for instance, in CLL, where you look at the doubling time of cells. I think we need to look at the dynamics of protein increases. What should be triggers that would allow us to anticipate that progression. So right now, most of the clinical trials are focused on high risk smoldering, and I should say smoldering remains a condition that we, under standard condition, would not recommend treatment. But boy, between the black and the white, there's a lot of gray in between. And I always say, teaching crab is a lot easier than living crab, because there's a lot of situations where we scratch our head whether someone has imminent risk of progression or not.
Well, so are you guys using the 22-20 rule in the clinic at this point, or the new IMWG project looking at sort of the weighted factor of that?
Yeah, I think I like the 20, the 20 rule, just because it's simple and it's something that we can incorporate, easy into the clinic, but it's not being used as of yet widely for treatment decisions. It still is an incorporation of everything else. I think those parameters and those frameworks are very good to look at in general groups that might be progressing. But I think that the question is there and will have to be addressed pretty soon, because as we know, there's now a second clinical trial looking at preventive treatment, so to speak. It'll be strictly called secondary prevention, perhaps, because someone has the disease, the early disease, and you're trying to prevent the progression to multiple myeloma. But the question is, who should be considering treatment right now for advanced smoldering? And I think, I like to think in general, when we do those determinations, it is more of a composite of even more variable. I think, in our particular case, we look at the growth rate, we incorporate the genetic factors. A key driver for me is the light chain, because the light chain is in and of itself is the highest risk for damage to the kidney. where we have a huge gap still in this predicting ability for smoldering going into active myeloma, is that we just don't have good markers for bone destruction. And we and others have been working on that. I think it's very, very early, sort of in diapers, so I can see that changing quite quickly.
Well, and if we look at that, right, so when you talk about, there's so many different treatments in the smoldering setting now, right? Mm-hmm. Vaccines, monoclonal antibodies, right, Revlimid, when you look through this, how are you viewing it how should a smoldering patient view it, maybe be a better question, with all these different treatment options, right? And then you kind of have the sledgehammer side, right, where it's taking Dara, Carfilzomib, Revlimid, and Dex on the essential. Where do you land on that? Is that too aggressive? I know we don't have data yet to totally understand this, but what's your thought process?
I do think, and this is just my opinion in August of 2019, right, that risk matters a lot when it comes down to the genetics, because that ultimately dictates what you do with the treatment. I think for a patient who has standard risk disease, who doesn't have any of the traditional high risk markers, consideration of what you might do, for instance, with early therapy, perhaps mostly focus on IMiDs, focus on the lenalidomide, right, might be appropriate. I think there's a school of thought that if somehow you perturb the microenvironment of the bone marrow, you might create an opportunity for emergence of more aggressive clones. Which in myeloma, it's possible, but it's largely a hypothesis. So there's no empirical proof for that as of yet. But it potentially makes sense that could happen, right? But when you have high-risk disease, then I think it's important to actually, if you were to start treatment, or if you're right there in that boundary that you don't know if treatment should be started or not, to go full force. And the reason for that is that IMiDs are not enough for high-risk myeloma. There was a recent abstract at last year's ASH stating how clearly that the mutational pattern would tell you that I think we're going to have to be using both proteasome inhibitors, and I'm pretty sure we're going to use monoclonal antibodies for high-risk disease. So I think that's how that's going to change, because the heart of the question is, if you decide to treat and you're uncertain, do you go full force, or do you just kind of lower somehow the risk of that progression? Now, based on what I said before, that I think is largely a probabilistic thing, a stochastic thing, I think if you have hyperdiploid smoldering and you bring down the cells to 10% of their level, I want to think, I don't know that, but I want to think that probably reduces the risk of that progression by a substantial amount. And of course, that's what the clinical trials suggest now.
Now, if we look at going from smoldering into myeloma, one of the key questions I think that comes up most often is, what is the role of transplant
Sure.
at this time? And sort of two questions. So what is the role of transplant, and what is the role of sort of what we call transplant plus? So looking at some of these new immunotherapy options or combining transplant with Dara or Carfilzomib or vaccine as part of the maintenance. What's your thought and what's your take on that right now?
Well, I think, so if you take it one step forward where you go, okay, the person is going to start treatment, it is very clear we're moving towards the idea of cure more than control. Now, I'm going to be very careful. I want to qualify that, because the first thing people will read is myeloma is incurable. I don't think that's true. I think there's a fraction of patients, most patients don't right now, but there's a fraction of patients that potentially can be cured with myeloma, but they can only do so when they go through more comprehensive and intense forms of treatment. And by intense, I don't necessarily mean intense against the person, intense against the myeloma cells. So right now we're going for someone who's going to go through a transplant triplet, stem cell transplant, and then they go with maintenance. That's kind of the standard approach. But where the field is moving, we saw a glimpse of this through the Griffin trial at the ASH meeting, where it's going to be 4 drugs with the Dara, VRD. It's not hard to anticipate it's going to be Dara KRD as we move into the future, and then a more tailored approach to your maintenance. What I mean by that is obviously standard risks remains Len, but would it be Len daratumumab? And the clinical trials are ongoing for that, of course, too. Even Griffin has that. We use Len plus ixazomib, then Inlaro for patients who have high risk disease because of some of the data of proteasome inhibitors. I know right now it's in trouble and there's a lot of discussion, but it's for me, it's not hard to imagine that in the future there might be a role for something like venetoclax maintenance for 11-14 patients. And the next set of questions is going to be, okay, should it be transplant or should it be some other therapy where, instead of transplant, maybe it's CAR T cells at the moment of minimal residual disease, whatever that means post-induction, or maybe CAR T cells rescue post-transplant for anyone who has MRD positivity. But I think the goal is to bring down those cells to as low a level as possible, hopefully to undetectable in the context of doing so safely for the person.
And there's so many questions I think that come up just from that statement, right? And I think it's going to be so interesting to see what happens on the CAR T side is that moves closer to upfront treatment, right? What are we do when we look at it at a fairly highly pretreated population? What happens is that gets in the second line and first line, and how does that change the overall response and the duration of response? But one of the things that when you're talking about transplant going into maintenance, where do you look at using consolidation? Do you have sort of a rule of thumb? Where does consolidation play into before you'd move into that maintenance side?
Yeah, that's a great question. No one has the answer. If you take the purest approach, you might say the clinical trials that have looked at consolidation have not shown necessarily great benefit, although some of them, like for instance, there was a Italian study looking at consolidation post-transplant with bortezomib. Thalidomide was better than thalidomide alone in the high-risk patients. Now, one thing that struck me, and I think one of the concepts that is going to have to be explored in future trials, there was a paper in September of 2018 by the French group that looked at MRD. And not until recently, I discovered that in the supplemental material they published, that if you reach MRD after transplant, or if you reach MRD during maintenance, the outcomes were very similar. So in other words, it doesn't matter that you may not be MRD negative post-transplant. There might be some benefit in still trying to pursue MRD after that, whether that is with maintenance or with some form of a consolidation approach. A little bit of this is semantics. Maintenance is kind of dialed down treatment, right? But at the end of the day, still anti-myeloma treatment. So I think as MRD becomes more and more mainstream in how we're measuring response after transplant, I think there's going to be more adaptive approaches to this, so that if you're MRD negative, maybe LEN is fine. And in fact, it might be so good that you might be able to discontinue LEN at a fixed period of time, because right now there's a school of thought of just keep going with maintenance, right? But if you're positive, then you might need to get very intense, because one is we know there's cells in the background, and number two is that might be the best opportunity to get rid of them. And the complicating factor, number three, is that we know that some of those cells could be just dormant MGOS-like cells. I mean, we see that all the time with patients who may have a small protein and remains there, and we just don't have the tools to tell A from B.
Well, on the MRD side, that's a good transition here, because if we're looking at that, the question is, how does that change maintenance, right? And that's kind of what you're alluding to, or what
Sure.
you're discussing here. I guess the question is, what does it mean, right? So if let's take a 1 in a million, or let's say your MRD number is 6 out of a million. What does that mean versus 0 versus 15? And again, maybe it comes down to the trend, right? What does that trend line look like? But if we look at this, is that a question that is being asked right now? If you're at 5, could you still do the same thing if you're at 0? And how are you differentiating patient populations to use this one number to determine discontinuation potentially of maintenance, or jumping to something else to maybe better control and then go to maintenance?
Sure. I think it's fair to say that until recently, MRD has been considered more as a qualitative state more than a quantitative one. So MRD positive or negative, and then kind of you get a glimpse of the number. But I think the next step is we need to move to that ability to be more reliable. Okay, when are we going to test MRD, and what samples are we going to send for testing for this? We just standardize this at our institution, where when we say we're going to do monitoring for MRD, the very first draw is what's used for the assays for MRD. So we try to have some ability to quantify what that MRD is, because I think you're right. I think the trend matters. And if you were MRD positive at 300, and after 4 months of consolidation you were down to 2, well, the question would be, should that person keep going, and should you try to still gain some more ground as you're going through this treatment? Likewise, if someone was at 20 and then you have 6 months later 300, I think everyone would be pretty concerned about the possibility of just being a matter of time before the clone reemerges and the person has symptoms again. So we need to get a little bit more standardized on how we're going to be measuring for quantity purposes the MRD measurements.
Probably standardization and getting some of these trials in to see what is actually happening on it. Like, so we're looking at some time to really begin to understand. I mean, years and years and years before we're getting a fuller, quantifiable, answer to that question, right?
Right. No, and I think you alluded, so you and I were having a conversation before. If people are interested, we have another video where you talk extensively about MRD, but I think the clinical implications are pretty interesting because I use the example of maintenance. People will say, well, there's no data for this or that, but here's how I walk through the logic. Some people would say it's perfectly fine to use 1 year of maintenance. Some people do 2 years. Some people will go forever. Keep going with your maintenance until the person either has no further evidence of disease control or can't tolerate for whatever reason or cannot have access to the medication. So if we accept those as reasonable options, the way I bring it is like MRD can help us. If you already had a year, you could have stopped anyway. So, there would be no criticism in someone who just gives Len maintenance for a year, at least not that we know of right now. We don't have the data. So if someone, at 1 year did MRD testing and they were positive, would perhaps the patient be more willing to take more treatment? Possibly. It would be just one more bit of information. Not necessarily 100% determinant, but it would be a bit more information. And if 2 years out, you're negative and, you're getting more fatigue or you're experiencing diarrhea, maybe that would be the extra bit of information you might need to say, I'm done with maintenance. And by the way, I'm very happy because 2 years out, I'm still at an MRD negative status. So I think that's how, we really need to push this agenda. It's a theme you and I have discussed, several times already during the conversation, the nature of dynamic monitoring and changes, as one goes through the disease process.
Yeah. Well, in being cognizant of time, this next one could go deep, right? So, okay, I want to jump into a CAR T
Sure.
side of things. Where we, can we talk about the state of CAR T? And we always say this is going to turn into alphabet soup, right? Because you hear BCMA, you hear CD38, there's CD138, SLAM-F7. So for anyone listening, it's not necessarily important to understand each one of those, other than knowing that right now there are different targets that are being considered or being used in the clinic.
Sure.
On the CAR T side. What's the state of this? How should patients and how should anybody be looking at making a decision, right? So if you look at all the CAR Ts that are out there with these different classifications or different targets, and we know that BCMA, B-cell maturation antigen, is one of the largest, right? It's the most prevalent at this point. How do you make decisions based on this?
Sure. Well, first of all, the CAR T cell field is obviously expanding rapidly and is here to stay. CAR T cells are essentially the next generation of cell therapies as a treatment for cancers. CAR T cells are really the perfectioning of what previously was attempted with allogeneic stem cell transplant. With allogeneic transplant, the whole thought was that let's give T cells from someone else, recognizing that they're slightly different from the recipient, just so that they can recognize those cancer cells as cancer, and of course remains an effective treatment and something that is used for leukemia. But really, CAR T cells are the designer product. The idea of creating those anchors against the various targets, and of course, as many know, we have products approved for lymphoma and leukemia, but myeloma is right there, and hopefully within the next couple of years, we'll have some commercial products for this. When we talk about these targets, of course, BCMA is the most common one, but there's others, the CS1 or SLAM-F7, and there's other protein markers that have been proposed as potential treatments via CAR T cell. What you want to make sure is that whatever anchor is being used has enough specificity so that the effect of this toxic T cell activity becomes restricted to the myeloma cells. Now, there's an aspect that we are fortunate, I'm not sure it's the right word, but we're fortunate in as we treat myeloma, and that is that the normal counterparts to myeloma, the late B cells, they are dispensable. One can live without it. It's not ideal. One has a propensity for infection. But if that marker is localized just to those very late B cells, and they're very effective, and as innocent bystanders, they take some of the other cells, it's not desirable, but it's something one can live with. So that's an advantage we have over the idea of developing CAR T cells that might recognize something else in the body, create an extra toxicity, Or even in myeloma, if some of the CAR T cells were to show some activity of what we call off target, that could be problematic. In fact, when BCMA, when AMG 420, which is a bispecific for BCMA, was presented, there were some glimpses of other toxicity that may or may not be off target. In that particular case, there were some cases of neuropathy, which was completely unexpected. So we're going to have to pay attention to how each one of these targets plays out. Going back to the specific BCMA, so BCMA turns out to be one of the best targets for the anchoring for those CAR T cells. It's one of the best specific targets. Obviously, that's why there's so many products. I think the products differ in how the constructs are made and how they create the co-stimulation and how they engage the T cells. And there's a lot to be said of how that may or may not matter in the very end. Things that we don't normally think of when we think about small molecules or other compounds, but how tight is a T cell against a target cell, whether there's exclusion of other molecules that may be immunosuppressive, whether there's enough engagement so that, it's going to create the cell killing effect. And at the end of the day, that's what we're waiting for, is just the empirical demonstration of which one of the CAR T cells is really going to be the best product. We're very excited. I think there's an overall perception, and you mentioned this before, that maybe the effect of CAR T cells in myeloma is not as durable, or has not been as durable as what we have seen in some of the cases with lymphoma. But people should remember, when CAR T cells are used in myeloma, these patients have, as you mentioned, 7 and 8 lines of treatment. So the T cells that show up to work, they've been through a lot. They've been through chemo, and they're all beaten up. And I don't think they can have the same health as the CAR T cells that are used in lymphoma, where a CAR T cell therapy may be their second or third line of therapy for a person with lymphoma, where at least hypothetically they have a better immune system. So I think as CAR T cells continue to be developed and move forward, they will be an option for myeloma. I think the toxicity seems to be somewhat different from what we see in lymphoma. And I always tell my patients, we're kind of ironing all the wrinkles so that maybe CAR T cells will be an option even for the very elderly, who maybe not normally be considered safe for a stem cell transplant, but might in the future be eligible for CAR T cells.
Well, and part of that too, when you're looking at that, the interesting thing is, so if we look at off target, right, some of these trials will allow for that kill switch or that off switch, right?
Sure.
Using what are typically either checkpoint inhibitors or some form of a monoclonal antibody, right, that can then deactivate that CAR T, right, to stop any off target, activity, right, which basically just shuts all of those down, right?
Sure.
But one of the points that I think you're making there is really about, maybe that's the play for the allo CAR T, right? The question here is, if there's an issue with our lymphocytes or the T cells or a patient's lymphocytes, does taking a donor-directed or donor-derived product help overcome some of those issues that exist there? And we're going to start seeing some of that in the clinic. In fact, there's two allo CAR T products coming up sometime in just the next couple of months across the US.
Absolutely, and I think for those in the audience who may not be familiar, so the CAR T cells are your own T cells, and the allo, of course, they come from someone else, and they have been engineered in such a way that you would remove the usual risk of graft versus host disease and those things. And I mean, they're quote unquote off the shelf, right? So for me, I think that's the biggest question right now, is whether those CAR T cells, as we're doing in myeloma, are going to stand the test of time, or whether it's going to be the allos, or for that matter, that may be a point of advantage for the bispecifics, because you can envision the T cells are infused back in the CAR T cells, and they're tired, and over time some of them either go away or lose their ability to attack the cancer cells. But with a bispecific, you're just reminding the immune system that this is something that needs to be fought off, and maybe it's going to be more effective. But we'll have to see, particularly as the usual CAR T cells, the autologous CAR T cells, are moved towards the front line of therapy.
Two sort of follow-up things. So number one, can you quickly touch base on venetoclax? And part of the question is, given the clinical hold that exists, right, which is if you're receiving the treatment already, if you're on a venetoclax, there's one phase 3 trial that has reopened enrollment. But if you were already on that receiving treatment, you can continue to, but no new patients are being added on. Is there a concern? Are you still prescribing that potentially off-label or using that in the clinic right now? And what concerns should patients have, and how should they talk to their doctor if that's being considered?
Sure. Well, first of all, I think, for patients who may be seeing this, they need to talk to their doctors, or actually seek a consultation with myeloma specialist if you're in doubt about what's the proper action if you're already on venetoclax. Obviously, there were some concerns with the clinical trial, with the Bellini clinical trial that had to be stopped, where it was tested, venetoclax was tested in combination with Velcade and dexamethasone. Parenthetically, there's a very good editorial by my colleague Vince Rajkumar that was published in Lancet Oncology just recently about this regarding the venetoclax. But I mean, the reality, I think this is still one of the most exciting medications we have for myeloma. But I think the key is going to be how do we position it, and specifically, that is, I think it should be, at least for starters, mostly be developed for the treatment of patients with the 1114. I have treated a significant number of patients already with venetoclax, which we started on treatment, some of them before and some even after the announcement came out regarding Bellini, because there's a number of patients who have the 1114 who have been refractory to other therapies, and we have treated them with venetoclax, usually in combination with some dexamethasone, where this has provided absolutely outstanding disease control. I think I may be seeing the first patient that is progressing on long-term venetoclax, but I have a fair number of patients that have been very well controlled. It's quite a dramatic response. Now, we use the 1114 as a surrogate for perhaps a group enriched for responses, and I fully acknowledge we don't know that that's 100%. There's some patients with 1114 who won't respond, and there's other patients that might, and people are looking at things such as the signaling proteins, BCL2, et cetera, to determine who might be the best candidate for this. We do some testing for the patient cells, and here, in our patients, when we collect a research sample, we can test it against the venetoclax and get a glimpse of what may be the activity or not. So I'm just hoping it's a matter of time before we go back to have this as one of the options for patients, even if it was highly restricted for now, to say you can only use it for 1114s is an incredible tool for these patients. And I ventured out to say, of course, we have to wait for some of the clinical trials. I think the editorial by Vincent points this out very well. But I mean, I don't think it's hard to imagine a future where venetoclax would be an integral part of frontline therapy for 1114s. Plasma cell leukemias, 50% of them have 1114. Light chain amyloidosis, 50% of them have 1114, maybe 1114 maintenance. And so I think there still are a lot of opportunities. We unfortunately do not have the full set of data to understand what led to the increased mortality that was seen in the venetoclax arm, but we know these patients were not selected. We know this was a trial in combination with bortezomib, and it was a global trial, so I think there's going to take some time before we get all the data to understand what happens. There's no clear evidence that this is toxicity related. Some of these deaths appear to be, in a later part or post-treatment. So I think we need to go down into the details. But when you look at patients with 1114 where we have used it, in my mind, it has been an incredibly effective drug. So I still go ahead and prescribe it for those patients that don't have any further options.
Prescribe it with antibiotics?
With that's not fully known. Initially, that was sort of the plan, and I think most of our patients are already chronically on antibiotics. They clearly are on, acyclovir, and the question is, what to do next. And so I know it's been the practice of many centers to prescribe antibiotics.
Okay. And last question, and this is actually, I think, a really important one. If patients aren't eligible for a clinical trial, let's say there's late relapse, not eligible for a clinical trial for whatever reason, right? Non-secretory, maybe a prior allo, some of those issues. We find that there's a lot of issues getting helping patients connect through compassionate use, right? And for anyone watching this, compassionate use is a way that your treating physician can actually petition a pharmaceutical company for access to a potential drug if they think it could be beneficial. And we find that a lot of centers will say we don't have the resources to run that. And I'm wondering, is that what you're seeing as well? Do you guys do a lot of compassionate use through Mayo? And the compendium to this would be, what about the right to try? Is there access to the right to try bill, which I'm sure you can explain, but how are you guys looking at, how at Mayo are you using compassionate use, and how easy is this to get there?
Sure. Well, we have used it, and it's interesting because even venetoclax for some time there's been sort of a blurring of what is sort of off-label versus compassionate use, because we can get it sometimes for patients from the manufacturer, if there's no way that we can have their insurance pay for it, just because it's been a useful drug. I think compassionate use has been employed over the years of, for access to some of those medications. And in fact, I think if one looks at the reasons why people seek compassionate use, they're perfectly good reasons. There's no other option. There's a presumption that a given treatment might help. I myself have been engaged in some of those requests, and some of them actually quite successfully, going to the company and getting the drug. I can think of an example. It's a little bit off topic of myeloma, but an example of a patient that was referred to me with something that's called the hyper IgG4 disease. This is a benign expansion of plasma cells, benign just because of the cells are not tumoral, but not because of the clinical consequences. This person had a deformity on the orbit already with the eye, had lost half the lung, was on the way to lose the rest of the lung, a life-threatening condition. So a physician referred her to me, and we ended up treating her with, at the time, Cyborg-D. So it was the Velcade, cyclophosphamide, and dexamethasone for a person who had failed everything. And that was 12 years ago, and she's in a complete remission since. And I'm not saying that this is an anecdote. This is obviously something that, or I'm not promulgating that this is the treatment for that. But for a person who was clearly on the way to lose her lung and potentially her life, where things were going, and now she's back to work and has retired and just leading a productive life, that's a great example for that. So we do participate, and I think whenever we think there's a need to do that, we have done that. We've even pursued emergency approvals through our IRBs and a bunch of other things. I think of an example where we tried to get an antisense therapy for a patient who was suffering from a West Nile virus encephalitis, that we had to get something emergently from California. And at the end of the day, it really doesn't even matter what happens. It matters of the intent and the understanding that the family has of what you're trying to do, and the patient understands that. And obviously, starting from this point of beneficence and that fiduciary responsibility that we have to the patient. Now, having said that, and you and I are talking about Sparkcures, and I'm committed to the whole process of clinical trials, but there's not always a clinical trial that is available to a person that could potentially benefit the individual. So you allude to this new legislation, that was enacted into law last year, the right to try, which essentially allows patients to bypass the regulatory requirements and just get directly medications from the manufacturers. I'll start by saying I'm actually fully supportive of that. I think it's completely aligned with patient empowerment, and particularly for something that may be life-saving or maybe life-prolonging. And as much as I talk about this fiduciary responsibility, and it starts with the whole concept of patient autonomy, I think you are in the best position to make decisions about your life, your future, what you put into your body. I'm your counselor, and I cannot force you to do things you don't want to do, or neither should I prevent from you doing things that you think are in your best interest. Sometimes I will disagree vehemently with what you want to do. I have examples of situations where I've seen patients who unfortunately have declined the treatments offered for myeloma, pursuing other forms of treatment, clearly with a much shorter survival. Patients with renal failure who went on to try some juices. Not the best decision, but at the end of the day, that is the person's decision, and that is where the right to try comes into play, because it's the principle of autonomy and self-determination. Now, we as physicians are counselors to patients, but if someone could have access to a medication this way, I'm of course fully supportive. I haven't seen this, how this would play out with biologics, but one of the things that Right to Try has is that it allows manufacturers to recoup some of the expenses associated with the production of some of these things. So I think that's where it's going to get interesting, because if you had someone who said, listen, I would like to have access to AMG 420 and I'd like to pay for it. Could I please have access to that, because maybe my creatinine is 2.5. I'm not eligible for the clinical trial. Is that a terrible idea? Probably not. I mean, if that's your last option for myeloma, it's not. Now, it is not like most bills, it is not a right. It's not a constitutional right, and the company is not forced or any way obliged to provide the medication. It's just simply that it clears a pathway for that to happen.
Have you ever used that successfully?
I haven't had to use it right now. I know it's something that probably will have to play out over the next several years. I'm aware of how people are trying to implement it, and some cases where it has happened. In myeloma, we're facing an interesting situation because we have had so many treatments that we're kind of there seems to be, I'm trying to think how to word this, but there seems to be a limit of how many things patients end up doing. So it is not sometimes that there might be nothing else to be done at the end of the line of a treatment for a person, but sometimes the patient or the family might say, it's enough. I may not want to go further, and that's also self-determination and autonomy, right? I recently had an experience of a patient that we were fighting to make her, and she was going to be eligible for a clinical trial with one of the bispecifics. But some introspection, and the patient said, you know what, I'm done. And of course, that was a moment of closure for her, for her husband, and and the kids, and that's a decision that was made. So I think that has come to play a little bit more in myeloma. But let's say if I was dealing with glioblastoma, I probably would be much more engaged right now in the search for opportunities in that regard.
That's helpful to know. We've tried it one time and it was denied. And it's interesting. So we're always on the lookout to see how people are utilizing that. They are edge cases, but when they exist, it's very acute, right? Sure. And you move as quickly as possible. So it's helpful to understand, your viewpoint and how you're.
And I always think things can be discussed both from the from the principle perspective and from the practicalities, right? And I think the practicalities are what play out over time. But I think the principle is very, very clear that I even if one person was helped by this, I think it's the right thing, because it allows you to be the master of your destiny, right? Whether the outcome is good or bad, it's that ability of me to respect what the choices you make for your life.
You get to select that level of risk, right? Knowing that it's maybe out of phase 1 and we still don't know a lot about it, but it presents another option, right?
Sure.
An option to help,
right? Well, and I should clarify, I forgot to mention, I think this would be helpful. This usually is for medications that have completed phase 1, that would be phase 2. Now, in phase 2, if I offer a clinical trial with a phase 2, which I, again, fully supportive of that, but if I offer a clinical trial with phase 2, I would have just as much information as someone who was pursuing the right to try. Because there may be 30 patients that have been treated in phase 1. And, if you had a toxicity that occurs in 3% of individuals, your statistical power to know that's happening would be very low with those 30 patients, right? So oftentimes it happens that, one goes to the phase 2, the phase 3, the post-marketing experience, things are kind of always in flux with regards to our knowledge about specific medications. So, Brian, let me give you a last word. I know we've been talking for a while, and we've covered a lot of ground. So thank you for your time. But it's really been a pleasure discussing this.
I'm so glad we got a chance to really get to, we run into each other at conferences and we cross paths, but it was really nice to get there and really get some time last week to really get to know each other. And, we're just really appreciative. I think that what I always like to say is thank you for what you're doing in your team there. It benefited my mom, it benefited my family. And I think that we all need to come together as a community, right? Healthtree, Myeloma Crowd, Mayo, all these great centers that are out there, and working directly with patients to help empower them is such a critical piece of this whole thing. So thank you for what you're doing. And anytime you want to do this, I'm more than happy to.
Appreciate it. I'm sure we'll have to do it more as this gets rolled out and we understand the clinical trial process. And down below, we'll put the links to both the Sparkcures, the Healthtrees, some of the other videos we have referenced to our conversation. And thank you for your attention.
Perfect. Thank you.